Well, welcome back, everyone. It's time for session 4, underlying cause of CF. So thank you to those of you here in the room. Thank you for those of you online. We've reached almost 80 people have joined us virtually, in addition to the almost 150 here in the room. I just want to let you know there's going to be an online evaluation form, an anonymous feedback form about the conference, which I'd really like you to fill out, please. So if you're here in the room, there's a QR code on your table. If you scan that, that will take you straight to the form. And if you're online, we're going to put the link in the chat box. So have a look in there. You can click on it and do that feedback survey for us. So this is our 4th and final session. We've got 4 excellent speakers. So first up, I'd like to welcome Dr. Paola Vergani from UCL and her presentation, Exploiting Fluorescence Assays for Screening Small Molecules and CFTR Variants. Thanks, Paola.
Hello, everyone. Thanks very much for the organizers for organizing this. So I always enjoyed looking at CFTR from up close. I used to be a patch clamper, but unfortunately in my lab nobody wants to do patch clamping because it's a very inefficient method and very frustrating. So I will be speaking about past research and future research. And one thing that people in my lab developed is a fluorescence assay to monitor CFTR. And it relies on the co-expression of 2 fluorescent proteins, a red fluorescent protein that is throughout the cytosol, and a yellow fluorescent protein that is fused to CFTR. And both are translated from the same mRNA. Now, the YFP is Galietta's YFP. And as you all know, it is anion sensitive. So when it binds anions, the fluorescence is quenched. But it's got a low affinity for chloride and a high affinity for iodide. So in the cell, we have the YFP on the cytosolic side. There is low chloride, so the YFP is bright. And in our assay, we activate CFTR with forskolin. So this assay was developed with a lot of work from Emily in my lab, Emily Langren up there. So once we've activated CFTR and it's at steady state, we add iodide extracellularly, and only if the CFTR is open and at the membrane can the iodide-chloride exchange occur, and we get quenching of the fluorescence. We quantify the CFTR activity by fitting the observed fluorescence quenching timeline with a mathematical model of the ionic fluxes. And we can estimate GCFTR, which is the whole cell conductance through CFTR. But we can also monitor the biogenesis of CFTR, and we do that by exploiting the M-cherry fluorescence, which helps us define the boundary of cells and also define a membrane zone very close to the boundary. And we can see how much fluorescence, YFP fluorescence there is in this membrane zone, and normalize it by the M-cherry fluorescence. Okay, so using this assay, we looked at, sorry, let's go back here. We studied the rescue of F508del by second site mutations. Now, first of all, I'll just talk briefly about the CFTR structure, CFTR up close. So you can't actually see the membrane in this slide for some reason. I'm sorry. There is a gray bar there. That's the membrane. But this is one of the cryo-EM structures of Ju Chen. Very beautiful. And this is another conformation of CFTR. This one is dephosphorylated. This one is phosphorylated. And here is F508. As everybody knows, F508 causes gating problems and biogenesis problems. And you can see here that there are two helices that extend from TMD2, which is dark gray, towards NBD1, which is in green. And F508 is at the surface of NBD1 at this interface with the TMDs. Now, these two helices form what is called the intracellular loop 4, and F508 is very close to that intracellular loop. Now, if we turn this structure 90 degrees so that we look from the side of NBD1, we can see that ICL4 dips down into a hollow like in NBD1, backed by ICL1, so an intracellular loop from TMD1. So here we can somehow look at the ball and chain, the ball and socket, sorry, ball and socket joints of CFTR. So there's the dip in NBD1, and ICL4 and ICL1 go into it. Now, focusing on this joint, Stella Prince, another PhD student funded by the Cystic Fibrosis Trust in my lab, asked the question, can two wrongs make a right? So if we have the F508del mutation, can we introduce a different mutation in ICL4 so that we rescue, at least partially, the defects of delta F? So what Stella did was to look at this systematically. So she did 5 or 6 substitutions at 11 positions in the ICL4 and studied this whole panel of mutants using our fluorescence assay. And these are the results she got for function. So you can see that the wild type, the first one in blue, has a high whole cell conductance of around 120 nanosiemens, while F508del in red is 25-fold lower. And then all these second site mutations were introduced. And what this result shows is clearly the R1070W was validated as an extremely effective revertant mutation. Next are 2 methionine substitutions at 2 phenylalanines at the interface between NBD1 and ICL4. And the assay also gives information on how much CFTR is in this membrane zone. And we can see that the R1070 mutation rescues both biogenesis and function. R1070W is the only one that rescues those significantly. Now, why is R1070W such an effective revertant? We did some studies of molecular dynamics in collaboration with Peter Tielemans' lab in Canada, and we came up with a hypothesis. So in the wild type, we have the aromatic ring from F508, and we have the arginine, R1070. The aromatic ring can form these hydrophobic stacks at the interface, while the arginine can form hydrophilic hydrogen bonds. Now, in the revertant, we have, okay, so now you can't see anything. Anyway, in the revertant, we have both the option of forming, we have a hydrogen bond donor in the amino acid of the indole ring, and we have an aromatic ring that can contribute to the aromatic stack. So both functions are replaced. Okay, so in the last few minutes, I will be talking about future research. So we were extremely happy to receive funding from both the CF Trust and the CF Foundation to study the bicarbonate fluxes in cystic fibrosis, the Restoring the Fizz SRC. Now, remember the fluorescence assay? We had a red fluorescent protein in the cytosol and the YFP fused to CFTR. We are planning to replace the M-cherry, the red fluorescent protein, with another red fluorescent protein that is also pH sensitive. So we are hoping to develop an assay that will allow us to simultaneously measure membrane proximity, ion channel function, and cytosolic pH. By altering the mathematical model to include also bicarbonate fluxes, we will be able to estimate both bicarbonate and chloride flow. Obviously, this is an extremely simplified system. These are hex cells. And in order to confirm anything that we find in our model system, we will be doing experiments with stem cell-derived material. In particular, we will use intestinal organoids and cholangiocytes and airway epithelia. And we will grow these to confluent monolayers, and then keeping conditions harmonized, we will be studying them using different techniques. So patch clamp electrophysiology, David Shepherd in Bristol, Ussing chamber measurements and pH stats measurements in Erasmus Medical Center, and my colleague at UCL, Guy Moss, will be using the scanning ion conductance microscopy technique to measure both the volume of secretion and the viscosity of secretions. So we are hoping that an iterative process of going from the simplified system in the hex to the more complex and more native systems in the ex vivo material will allow us to hone in on the most interesting aspects and hopefully comparing regulatory systems in different tissues, gain some insight in how the bicarbonate and chloride fluxes are regulated in these different tissues. So what will come out of it? We hope that the protocols that we will develop will be drug development tools for better modulators in the future. In the short term, by studying these different tissues and comparing the bicarbonate and chloride fluxes, we are hoping that our studies will also help guide clinicians in choosing which modulators individual patients could benefit most from. Okay, so acknowledgements, certainly the trust for funding. I really would not be alive research-wise without the CF Trust. And all the people that worked in my lab, in particular here now, Stella and Emily. But I would also like to acknowledge these new 5 PhD students that we have recruited for our Restoring the Fizz SRC, who are really excited to start their work. And I thank you.
Thank you very much, Paola. So I'd like to introduce Dr. Patrick Harrison from the University of Cork to give us an update on the Gene Editing Strategic Research Centre. Over to you, Paddy.
Thanks. Okay, just like to thank the organisers for the opportunity to talk today. My name is Paddy Harrison. I'm going to be talking about fixing CF-causing variants in the context of the gene editing SRC. So there's a lot more of us, and the 3 people in red here have got posters outside, and they'll all be talking in detail about their own research. So rather than me spoil that, I thought I'd talk more about the broader context of trying to use gene editing as a therapeutic tool. We've seen this slide already today from Jane, I think it was. This is the editorial from that issue of Science when the CF gene was cloned in 1989. And Daniel Koshland, the editor at the time, described the gene cloning as the very happy middle of a story, which ends with a cure. So I Googled cure for CF, and apparently there's quite a lot of interest. But the question is, can we actually deliver this using gene editing or gene therapy? People always ask, when will it be ready? This isn't a prediction, but that's how long it is till the end of this current decade. And is that a realistic target? Just something to think about. If you look at the cause, this is extremely well understood and very simple to explain. If both biological parents carry a variant for CF, and the embryo which is created from those parents carries both those variants, the person will end up with cystic fibrosis. But since it's a monogenetic disorder, there's only one gene which is involved which carries the variants, then if we could target that gene, then potentially that should be the basis for a curative therapy. And editing the gene has actually worked for other diseases. The girl in the first picture there, she received CAR T cells which had been gene edited, which saved her life. The lady in the middle, which I'll come back to a little bit later, Victoria Gray, was the first person to have her own cells genetically modified by editing and then put back, and that's actually cured her of sickle cell disease. And then the person on the right here has had her vision restored in vivo by using gene editing. That's just a quote from Woody Allen. Let's just look at the basis of this then to get some ideas what we're talking about. With the CF gene, we make a messenger RNA, it gets spliced, that mature message moves into the cytoplasm, it makes a protein, we get chloride transport. But if we get a mutation like G551D, the one which responds to ivacaftor, we make the protein, it just doesn't open properly. If we have F508del, it doesn't traffic properly. If we get stop codon mutations, we get a truncated protein or virtually no protein. And if we have splice mutations, we don't even have a protein to start with. The beauty of what's called a cDNA therapy is that if we could introduce a full-length cDNA, that should make a messenger RNA. And the really cool thing about this is, as we all know, this would fix any mutation. And this is why there's been so much interest in this for the last 30 years or so. But if we could do gene editing, if we could just have a magic wand to come in and correct that mutation, then we'd make a messenger RNA, which is exactly the same sequence, except it's under the control of the endogenous promoter, and it will be expressed at the right level in the right cells throughout the body if we could do that. So let me just show this slide. Normally we see gene therapy in the blue corner and gene editing in the red corner, and it's going to be a big fight. Well, I think that's rubbish. I think these two technologies need to be used together because we can learn so much if we combine them. The ultimate goal is to restore protein function in as many cells as possible in the right tissues, and obviously that then translates into potentially a highly effective therapy for everybody with cystic fibrosis. But it all starts with the same route. It's quite a busy slide, but we've got cells down the bottom which we grow in a tissue culture flask, and we've got a whole variety of different delivery systems which we could use to deliver a cDNA or messenger RNA. Ultimately, though, the goal is to get them into the lung of people with CF initially. And people have been trying this for 30 years. This is data from the first clinical trial from nearly 30 years ago from Ron Crystal and colleagues, and it was safe, just not very effective. This is the data from the UK CF Gene Therapy Consortium from about 7 or 8 years ago. Again, very safe, shows that you can perform large-scale clinical trials with all the logistics which goes with that, but it's nowhere near as effective as modulators. But that doesn't mean that people haven't given up trying, and it doesn't mean that people haven't given up hope. There's at least 7 companies which have got CF gene therapy using cDNA or messenger RNA in preclinical development, and there's at least 6 which have started clinical trials or are just about to start clinical trials. And I just want to show 2 slides, one from each of 2 of them. This was a study which was published by Steve Rowe and colleagues in the last few months, showing that they tried to deliver messenger RNA for CFTR in the lung for the first time. And as you can see, I've highlighted it was safe and well tolerated, but there's no major improvements. The second study, which is also reported this summer at several places by 4D Molecular Therapeutics, shows for the first time these little black blobs, which you can just about make out there, are evidence of expression in the lung. So they don't have data yet to show whether this is effective. This has only been done in 3 patients, as far as I'm aware, so far. but it's giving hope that we can deliver, or the community can deliver, messenger RNA or cDNA, I don't believe that's my cue, to the lung. Now, what can we do? Can we use that information about delivery to help with editing? Well, everybody thinks editing is a brand new technique, the Nobel Prize 3 years ago, but it's got its roots back in the 1980s. Very inefficient, a double-stranded break can help, but we didn't get the tools to make a precise double-stranded break until about 15 years ago. We were one of the first to use this technique to correct the mutation. It took a long time with zinc fingers and some funding from the CF Trust. But when CRISPR came along, this just revolutionized and opened up the field so that virtually anybody can try this approach. This is a slide from Jeff Beekman's group. The little green blobs here are organoids, which you're all very familiar with as a way to screen for drugs. But when you take edited organoids, these also swell up. And that should be no surprise, because once these cells have been edited, they're effectively completely wild type. For many years, we all had a lot of fun doing editing until Bill Scatch made this statement at the North American meeting just before COVID. New developments in gene editing must be amenable, American pronunciation, to clinical development. And what he was getting at there is we can't just do this to see if we can fix mutations in cells. We need to get this into a strategy which is compatible with use in people. But we've seen remarkable progress in the last couple of years with gene editing in people. This is a study which was described for this rare liver disease. And what you see in the blue blob in the middle there is a messenger RNA, same kind of thing which we've been shown to deliver to the lung, and a guide RNA, which when they get into cells, can produce protein. So they've been introduced intravenously, that's in a capillary. We zoom down to the liver, which is their site of interest. And as you can see, just over my shoulder here, you can see the protein has now been assembled. That protein RNA complex can move into the nucleus and then bind to a specific bit of DNA. And this is the critically exciting part about CRISPR gene therapy. You can target this molecule to a unique site in the genome, very close to the mutation that interests you, and then you can use different strategies to correct it. My lab used this strategy several years ago just to make a very simple strategy by cutting a deep intronic mutation. Other labs showed a similar strategy in this restores function, and in our first SRC, we've now just got the paper coming out with several others on it, showing that, again, further details of the restoration of function. But that's just one mutation. The reason I'm showing this is because the same approach has been used in this disorder, and it's actually gone as far as a clinical trial and restored the vision in at least 2 people. But a non-clinical challenge that we face is that it's going to be very difficult to raise the money to pay for all of this. There's lots of different strategies. We're using all of these in our SRC just at the moment. There's always something new with CRISPR. There's been 2 publications which have really caught our eye just in the last few days. In terms of delivery then, just let's explore those 2 options. We can learn from gene therapy. If we can see more of this in the clinic, if it doesn't work out from a cDNA-based therapy, which I genuinely hope it does, but if it doesn't, we could exploit this for gene editing delivery. And with the systemic delivery, we've seen targeting to the liver, but if we just change some of those proteins in the red boxes, maybe we could use a systemic approach for targeting the lung. There's a lot of questions on this slide, which Lucia, Izzy, and Miriam will answer in about half an hour's time, hopefully. But I just want to finish on this slide. This is just data from CFTR2. 140,000 alleles, about 70,000 people. You can see there's a lot of alleles which don't respond to modulators, but when we do the numbers and put this into people, you can see the numbers actually very small. That's very good, but it also makes clinical trials for gene-based therapies very difficult because the global population is very small. Now, if you're doing an mRNA trial, there's 3,000 people eligible. But if you pick a specific mutation, W1282X, which many of you recognize is actually relatively common, then there's only about 350 people worldwide who would be eligible for that trial, just in principle with the mutation. And finally, I said I'd mention Victoria Gray again. I heard her speak earlier this year in London at the CRISPR Summit. And when she was asked about her enrollment in the trial, she said, to the clinician involved, what would happen if it doesn't work? Could I still get a bone marrow transplant from my brother? And once she was reassured that she could, she said yes to the trial. The ethical question that we face in CF, and James mentioned this morning, is will a person with cystic fibrosis be able to get repeat doses of certain genetic therapies and or take part in trials for other genetic therapies. Okay, I'll stop there. Thanks very much.
Thank you, Paddy. Next up, we have Professor Robert Gray from Glasgow University, who's going to be presenting on CF lung disease from inflammation to repair. Thank you.
Thanks. Okay, good afternoon. Oh, I see my axis has disappeared. Good start. So, my name's Robert. I'm from Glasgow. I used to work in Edinburgh for many years, and much to people's surprise, I left and went to Glasgow. Usually happens the other way around, but there's a good reason for that, because there's lots of good stuff happening in Glasgow now. So I'm going to talk about inflammation, and then potentially how we can go from inflammation to tissue repair, which is a really big subject for 10 minutes. But the first thing to say is that, or why is inflammation important in CF? Well, it's important because we know that people with higher levels of inflammation have a worse clinical outcome. Here we're looking at the serum marker calprotectin, and people with a higher level of calprotectin exacerbate much sooner than people with a low level of calprotectin. And we know that there are certain things associated with CF that are specific to CFTR function in terms of inflammatory problems with immune cells. So we've spent quite a lot of time over the last decade looking at immune cell function in the context of patients with CFTR mutations, and it's really quite stark to see that you can only I can only get about 4 lines in large font on one slide that shows how much work I've done in the last 10 years. So anyway, we've shown that the neutrophil apoptosis phenotype in CF is different. So CF neutrophils have delayed apoptosis, and neutrophil apoptosis is normally a mechanism of inflammation resolution. We've also shown a pro-inflammatory macrophage phenotype in CF, so CF macrophages produce higher levels of pro-inflammatory cytokines. And we've also shown that there are CFTR-dependent dysregulation of certain pathways, such as interferon signaling, and we can correct that by editing CFTR. So suggesting that there are distinct problems with CF immune cells that are related to having a deficiency of CFTR function. And we think that the interaction of neutrophils and macrophages may amplify inflammation in CF. And to put it more simply, bad neutrophils and bad macrophages lead to bad inflammation. And we can also think of bad inflammation in the context of it being frustrated inflammation. So we all have inflammatory responses to vaccines, to pathogens, and usually we have a very quick immune response, which then goes on to resolution, and it's during that resolution response that we would classically see tissue repair beginning to happen. However, in the context of CF, we never really see that resolution. What we see is a sort of protracted acute inflammatory process that never really gets any better and is characterized by lots of neutrophils and lots of pro-inflammatory cytokines. And that's what leads to this destructive lung disease that until now has been characteristic of CF, but may of course be changing with the newer treatments that we have available to us. So a major question that we wanted to answer was, if we corrected CFTR dysfunction, would inflammation improve in our patients? Or we wanted to make it more complicated and say, if inflammation improves in CF, can damaged airways repair themselves? Now, I do know that I've used the word massive and huge here, which may be a bit hubristic, but I suppose the point I was trying to get across was, this is a really big question to answer, and it's really difficult to think of even how we would start with answering this sort of question, thinking about a lung regenerating itself. So we were very lucky in that a local charity in Scotland, Warren's Wish, gave us a small amount of money, and the CF Trust gave us a VIA to allow us to supplement that income. And that enabled us to employ 3 very bright young people, Jonathan Gillen, a PhD student, Gareth Hardesty, a postdoc, and Nicola Robinson, who was a clinical fellow. And this money allowed Nicola to be employed for a year and then gain an MRC training fellowship. So it was really useful. A small amount of money led to a massive amount of progress for us in this area locally. So I'm going to now show you a bit of data about the sort of things we've been addressing in our project so far. And all of this data really has been generated by Nicola and Gareth and Johnny during their time in the lab. So does inflammation resolve when we improve CFTR function with highly effective modulators? Now, we, of course, are not the only lab trying to answer this question, but this has been our approach to it. So Nicola did a clinical observational study where she took samples from patients before and after a highly effective modulator. And this first slide here is to show you that actually, when we looked for changes in inflammation, we expected, or we hypothesized, we would see lots of different inflammatory mediators changing. And this was a test set and a proteomics analysis of patients who were completely naive to modulator and then had 1 year of treatment. And as you can see, it's the sort of Jackson Pollock of heat maps. There's really not much of a signal there before, or a signal there at all. If we drill down into the data, really only 3 things significantly changed in this proteomic analysis, and those were IL-6, MMP10, and IL-20. When we looked at this in our larger group of patients, about 40 to 50 patients, we could confirm that IL-6 had improved in these patients after treatment, and we also, calprotectin wasn't in our original proteomics analysis, and we were able to show that that decreased over the course of treatment, so suggesting that there was a change in specific aspects of inflammation with treatment. So, some aspects of inflammation get better, but what does that mean for patients? Was there any impact on structural lung disease? And Jane already showed some data or some pictures today showing the really quite obvious and stark changes we get in patients who have had treatment. But this is an adult with established CF lung disease. This was during COVID, so we only managed to scan 10 patients who were completely modulator naive before and after a year of therapy. And as you can see from this patient here, in the pre-treatment versus post-treatment scan, there's really naked eye significant changes in the amount of airway wall thickening and just the general lung architecture. And then if we look, this is in a patient with slightly milder disease, but this is just to give you an idea of how we've not just chosen one slice of one particular scan. If we look at patients treated with these drugs, we're seeing a decrease in inflammation that is really manifest in terms of what we see on a CT scan. And we've looked at these CT scans of these 10 patients with expert radiology scoring and also with AI, although we probably should have just used actual intelligence and looked at the scans, and we would have saved several £1,000s on the analysis. But the impressive thing is that we see not just bronchial wall thickening improvement, but also improvements in bronchiectasis and improvements in even size-significant mediastinal lymphadenopathy. So major impacts on inflammation that have a cut-through into patient radiology. We seem to be stuck. Okay, so from this part of the talk, really our conclusion is that better CFTR function is associated with less inflammation and improved lung disease. We see a sustained reduction in specific biomarkers with CFTR modulation, and that is suggestive that there may be some degree of inflammation resolution. And we find that this is associated with significant improvements in lung disease on HRCT. And the question I'd like to pose is, are we seeing the early stages of lung repair in our patients? Now, that might be a fanciful question. However, I think that the data we've shown so far in terms of the CT changes is really promising that something significant might be happening. So the last question I was going to leave you with today was, what does modulator therapy do to the cells involved in tissue repair? And this, again, is work that Nicola and Gareth and Johnny concentrated on. And everyone here will probably be aware that we have a resident stem cell population in our lungs, or a resident population of cells that can act as stem cells. These are basal cells. And basal cells respond to injury when it occurs, migrate to the point of injury, and can differentiate into individual cell types. We know from previous studies that there is basal cell hyperplasia in the CF airway, but the basal cell hyperplasia does not lead to a normal airway being repopulated. What we see is a disorganized epithelium with a loss of ciliated cells and more secretory cells. And when we look at single-cell sequencing of air-liquid interface cultures that Gareth did with a collaborator, Malcolm Broadley in Newcastle, we could see in our CF patients they actually had more basal cells. So they had basal cell hyperplasia, but this basal cell hyperplasia is not leading to better epithelial cell repair. So one of the things that Nicola looked at in our patient group before we had used modulators was to compare basal cell function between CF and healthy controls. And she took nasal brushings, and here we're looking at nasal basal cells rather than basal cells directly from the lung, but they're very reflective of the basal cell population in the lung. And what Nicola showed was that after expanding basal cells in culture, CF basal cells have a reduced rate of proliferation, and also when she used these cells to form organoids, healthy control organoids formed nice-looking organoids that would be associated with the sort of things you would see in a publication, whereas CF formed these very small, irregular-shaped organoids that looked very unhealthy. So our next question was, what would be the impact of modulator therapy in this setting? And here Nicola's taken basal cells from nasal brushings in patients who have taken 3 months of modulator therapy. And what she showed was that actually in patients who had been on therapy for 3 months, when we took those basal cells out of the nose and cultured them, they actually had a significantly different transcriptional profile. And lots of genes were changed after that 3-month treatment period. And several of the pathways that came up on ontology analysis were pathways that would be associated with tissue regeneration. So I'm going to finish. I've just got 2 more slides, if that's okay. Nicola then went on to check whether whether the effects on basal cells were due to a direct effect of CFTR modulation on basal cells, or whether it was due to something else. And what she showed in a nutshell was that adding triple modulator to ex vivo cultures made no impact whatsoever in terms of the basal cell dysfunction. So it had no impact on organoid formation or even on a simple scratch assay, perhaps because we would not expect it to be a huge amount of CFTR expression in a basal cell anyway. So conclusion 2, the repair machinery is not working properly in CF, and this might be due to environmental conditions. We know that ETI treatment now in our patients is associated with a pro-repair signal or signature, and these effects are not seen on basal cells when we add ET to ex vivo models. So we think it's likely an effect of the ETI and the decreased inflammation in the environment where we're taking the cells from that's having this change in terms of the transcriptional profile. And this leaves a question, or several questions. First of all, is improving inflammation driving better tissue repair? Could the interaction of immune cells with basal cells be a key target? And can we now model this ex vivo using primary cells? I'm going to finish by just showing you this last slide. This is what we've been doing recently. You can tell that this is a real experiment because it looks terrible. And this is a simple model where we're using ALI culture with a fluidic support, but we're hoping to develop this model to be more complex, so to involve matrix and fibroblasts and endothelial cells, etc. So to give us a model to be able to discover what might be happening during that injury and repair period and how that might be different in CF. And I'll finish there. Sorry about going over time.
Thanks very much to Robert. So our final presentation before the Q&A is I tried so hard to learn this. Deubiquitinases as therapeutic target, modulation of ion channel function in the CF airway epithelium. So please welcome Dr. James Reihill from Queen's University, Belfast. Thank you.
Yeah, okay, thank you. So maybe I'll just call them dubs for handiness. But so yeah, I'll just dive straight in. Oh. I'm missing the first couple of slides. So I'll have to, well, I'll see if I can try and remember what they actually are. So the background is in terms of airway hydration and mucociliary transport. Okay, so we know that correction of CFTR can improve airway hydration and mucociliary clearance. And that's suitable for some, but not all patients. We have other alternate ion channels present in the airway epithelium, including sort of TMEM16A and epithelial sodium channel, or ENaC. So they provide sort of other routes to restore airway hydration and mucociliary clearance as well. And so these ion channels can be targeted directly, for example, with sort of potentiators, correctors, channel activators, blockers as required in terms of ENaC. But they can also be regulated sort of indirectly, which leads into the concept of proteostasis, which is a process that regulates proteins within the cell to sort of maintain a healthy environment. Okay, and this cellular environment can be targeted in order to indirectly regulate ion channel function. And proteostasis regulators include members of the ubiquitin proteasome system, which has a central role in protein degradation or recycling of ion channels. So essentially you have a deubiquitinase, which allows ubiquitin to a protein target and signal it for proteasomal degradation, whereas a deubiquitinase or DUB can do the opposite, remove a ubiquitin and sort of facilitate recycling of that protein. And shown in the figure in the bottom left, you can see sort of several sort of targets for CFTR proteostasis regulators, and within that you see sort of the DUBs or deubiquitinases. And really there's only a few papers in that regard. Some look at sort of USP10, USP19, how they can influence CFTR. So really the DUB family as a whole is quite poorly understood in how it can regulate channels like CFTR. There's some DUBs linked with ENaC and really very little with TMEM16A. So, but really sort of quite an incomplete understanding of DUBs as a family and the regulation of these ion channels in the airway epithelium. And quite fortunately, sort of co-localised the same life science campus where I'm based, sort of in Queens, there's a company, Almac Discovery. And Almac has significant expertise in targeting the deubiquitinase enzyme class, mainly sort of for oncology programs of work. But we felt there was opportunity here to bring sort of a combined expertise and look at the role of deubiquitinases within sort of chronic airways disease, including CF. Oh, there's my first slide. Let's skip. That would have been a lot easier trying to remember. So if any more disappear, I'll flick through. Right, so to try and get this sort of project off the line, I was fortunate in that I was able to receive this sort of Innovate UK Innovation Scholarship. And that facilitated secondment to Almac Discovery, where I was embedded in the company for 18 months, which is really, really useful experience. So it was really designed in terms of sort of career mobility, as well as knowledge exchange between academia and industry, and covered sort of the salary and subsistence and various things. And then the CF Trust VA award scheme was able to then also support the program as well, and was crucial in getting this sort of off the ground. So we had two main research questions. One was, so do members of the DUB family modulate activity of these ion channels, ENaC, CFTR, and TMEM16A in their epithelium? And if so, can we target or manipulate those in a way to improve airway hydration and mucociliary clearance? So to address this first question, we formed this DUBome or DUB-wide screen. Essentially, we grew up, these are non-CF human bronchial epithelial cells, and then carried out sort of an arrayed CRISPR-based gene editing screen where we knocked out 94 individual DUBs, and then grew these sort of an air-liquid interface for 4 weeks and measured ion channel activities using the TAC24 system. And then I'll just talk about one candidate that emerged of particular interest, which I'll refer to as DUBX, as we kind of still explore sort of the commercial opportunity with that target. And we were interested in it because not only did it increase CFTR activity by around 35%, but also had potential beneficial effects in terms of increasing TMEM16A by around 30% and blunting ENaC activity by around 20%. So we followed up those sort of knockout studies with a pharmacological inhibitor developed by the company. It's highly potent and selective for this target. And again, found similar results. We sort of increased CFTR activity, increased TMEM16A to a degree, and sort of perturbed sort of ENaC activity. And then we proceeded to evaluate the inhibitor in CF cells, first these sort of homozygous for the F508del mutation. So we did the experiments with and without our DUB inhibitor and with and without ETI, sort of to benchmark our responses and also to see if there are any additive effects. And if you look in panel A, we see that ETI increases CFTR activity, as you would expect. But in the presence of the DUB inhibitor, we enhance this by sort of a further sort of 35%. When you look at panel B, TMEM16A, ETI doesn't really affect TMEM. But when we have our DUB inhibitor in there, we increase it by around 1.75-fold. And then when we look at ENaC, we find that ETI or the DUB inhibitor by themselves reduce ENaC activity by around 10%. When you have them combined, you get about a 30% reduction. So we're seeing these sort of effects with our sort of DUB inhibition in terms of multiple ion channels. We want to know, what does this mean in terms of mucociliary transport when we affect this sort of multi-target approach? And so we form these sort of mucociliary transport assays, where essentially we're adding fluorescent microbeads to the apical surface of our alley cultures, and then 48 hours later, sort of evaluating their movement by sort of live cell microscopy. And sort of the more movement, you sort of track the movement and assign sort of a velocity of sort of microns per second. And then in the intervening period, we're adding our various treatments basolaterally, whether that's sort of the DUB-X inhibitor or ETI. And if you see sort of the figure in the bottom right, you can see that ETI increases mucociliary transport, as does the DUB-X inhibition to a similar or maybe slightly greater degree. And really you see this sort of large amplified response when you add both in combination, maybe sort of a 4-fold increase with the combination compared with ETI alone. So a few things we're thinking about, and bringing it back to sort of the JLA priority settings that we've mentioned several times today. One area of interest is really, this fits with sort of number 1, which I maybe can't remember exactly, but essentially talks about options for those that aren't suitable for CFTR modulator therapy. And that could include whether they just have sort of a genotype that isn't suitable, or maybe tolerance issues. So one thing we're keen to look at is whether we can sort of titrate ETI down to much lower doses, sort of with a complementary therapy like this, and whether that would negate some of the side effect issues. And then also, because we see effects of the DUB-X inhibitor alone, sort of this sort of in the absence of any CFTR correction, what effect will that have in sort of, for example, class 1 mutations where we have a loss of CFTR protein and thus there's nothing that's modulatable. So we've done just a little bit of work, some preliminary work in recent weeks, looking at some HB cells carrying this nonsense mutation. And encouraged, we do find there's this uplift in mucociliary transport in the presence of our DUB-X inhibitor. So we're keen to kind of look more at this in the coming weeks and months. So with that, just to summarise what I've shown you, sort of a non-CF HBE, we performed this sort of gene knockout screen of the sort of DUB family and identified DUB-X as a candidate that simultaneously modulated ENaC, CFTR, and TMEM16A, with sort of potential to sort of drive aerohydration and mucociliary clearance. In CF cells, F508del mutation, inhibition of the DUB-enhanced ETI sort of induced CFTR activity, and by itself boosts TMEM16A activity and sort of blunts ENaC. And this sort of then leads to sort of an improvement in mucociliary transport when added by itself, or even so amplified when added in combination with ETI. And then finally, in these sort of class 1 HBEs, we find that DUB-X inhibition by itself increased mucociliary transport, highlighting potential as a potential sort of mutation agnostic approach that would be suitable for all patients or all people with CF. Yeah, and so with that, I'd just like to acknowledge sort of the team, particularly those in Almac Discovery. They were very welcoming, and it was a very useful experience being within sort of the company. And then a special thank you to the CF Trust and the VIA award scheme that was critical in getting the programme up and running, and hopefully we'll be able to continue it and get some more interesting results in the coming months and years. Thank you.
Thank you very much, James. So let's bring Paola, Paddy, and Robert back up on stage, and we'll have a quick Q&A session. So Paula's going to be relaying the online questions. So Paula, I'll probably come to you first this time, just to alternate it. And you've got the catch box and the roving mic, so be ready for that, people that want to ask questions in the room. You're off the hook now. You're not going to have to catch it. Okay, so Paula, any questions from the online audience for our panel?
Yes, there is one, I think relating to the gene editing SRC. Do you have any candidate lung cell surface proteins for nanoparticle liposome delivery?
Okay, so I think the question relates to targeting the different liposomes. So Steve might be better at answering this question than myself, but his nanoparticles contain peptides which will target various different proteins. From the virus side of things, there's several candidate proteins which have been identified, and for some of our own work on virus-like particles, there's a receptor which is looking quite interesting. But the 4D molecular therapeutics one I mentioned there, their virus which has been modified there, they screened thousands, if not millions of viruses to try and do it without knowing what target to go for, but to look to see if it could deliver. So there are ways and means to achieve that.
Okay, thank you. Any questions from the room? Yeah. Throw the box at Professor Floater. Thanks.
Is it on?
Yeah.
Yeah, lovely. Question for James then. So the DUBX presumably has more targets than just CFTR and ENaC or TMEM16A. To what extent do you think that promiscuity may limit therapeutic targeting of that DUB?
Yeah, so it does have multiple effects. We've started doing some transcriptomic work to get an understanding of sort of the overall cellular biology. So that's really ongoing, and so we don't really have the data from that at the moment to understand. From some of what we've seen, though, if I do some qPCR analysis of the various ion channels, it looks like we see an upregulation of TMEM16A, not the other two. It's clear from the literature that there's potentially an interplay between, for example, TMEM and CFTR. If you knock out TMEM, you can lose CFTR activity. So one question is whether the initial effect is on TMEM, and that sort of is a secondary effect on CFTR, or there's interplay between CFTR and ENaC. So I don't know if it's it's just this inhibition of this is double doing loads of things, or it's actually more it's affected TMEM, and that's having secondary effects on those ion channels. But really we have to look more broadly at the sort of cellular biology. And we're also looking at other targets. I just picked one today. We have another one that's very CFTR-centric, another DUB, so we're actually looking sort of at both, where one's doing the sort of multi-target effects, and one is really doesn't seem to do anything at a transcription level, but does stuff to CFTR, so we're kind of looking at both approaches.
Thank you. No further questions online? Okay, so back to the room, if anyone has a question. Yep.
The question's for Robert. You mentioned the differences with apoptosis with some of the neutrophils or the macrophages. Increasingly, there's this idea of like aging cells or zombie immune cells that are hanging around when they should be dying and contributing to this sort of pro-inflammatory environment. Do you think that the actual modulators are actually making some of those cells die earlier before they're aggravating the environment as much as they could be?
Yeah, it's a good question. And if I'd had time, I could have shown you some of the data. But in our paper where we looked specifically at neutrophil apoptosis being delayed in patients who had G551D mutation, if we gave them ivacaftor, even after 3 days of treatment, when we then took their neutrophils and cultured them ex vivo, they were behaving more like healthy controlled neutrophils. So, and the other thing that we know about CF neutrophils is when they survive longer in culture, we can give them various stimuli, and they'll then go on to have different forms of cell death, such as netosis. So, I think that the evidence is there, certainly circumstantial evidence that perhaps modulators are doing things like changing CF neutrophils back to a more normal phenotype. And in terms of the macrophage data, when we looked specifically at gene expression in CF monocyte-derived macrophages in response to LPS, we saw lots of differences between CF and wild-type macrophages, and many of those were reversed when we treated ex vivo with a modulator. And when we corrected CFTR with CRISPR in iPSCs derived into macrophages, we showed that we reversed some of the signaling genetic signatures. So I think there's definitely an element that we have immune cells that are behaving abnormally. Whether that's the entire story in CF is another question, and it's most likely it's a combination of that immune cell dysfunction plus things such as infection and the mucus abnormality that leads to that perfect storm that's driving inflammation. But certainly we think that the immune cell problems are part of it.
Thank you.
Thanks. Okay, we've probably got time for one more question. I think we haven't got any questions online. Any further questions in the room? Okay, not seeing any questions for each other?
So, Paddy, tell me about the off-target effects of gene editing, because I was quite surprised when I went to a talk recently where off-target effects might be, or we were told in that talk, off-target effects might even be underestimated by the techniques we're using in terms of computational modeling at the moment, where things not just due to sequence, but even the shape of your guide RNA might have really quite significant off-target effects. What would be your feelings on that and your concerns as we go forward with CRISPR into CF as a treatment?
Yeah, so this is something that's happened over the field as it's matured. So in the early days when people were using CRISPR just to make a double-stranded break, it was highly effective for correction, but the first papers alerting people to the potential complications of this led to the development of base editing and prime editing, which rather than making a double-stranded break, make a nick. People have shown that these nicking Cas9s significantly reduce this, but don't eradicate it. But with careful design, you can bring it down, approaching the level of mutations that you might see just by cells replicating. There's always the potential for off-targets. One of the things we discussed at the American meeting this summer is when looking for off-targets, people use a white reference genome, which hasn't considered lots of ethnic diversity. And there's a really good example, unfortunately, in sickle cell, where a major off-target was when it was found when they started looking in genomes in populations which are much more affected by that. So you've really got to be careful. One topic that came up again in that meeting was that list I showed, there's only maybe 3,000 people with cystic fibrosis. And if someone was considering having gene editing as a treatment at some stage in the future, maybe sequence their entire genome if they were interested, do a pre-screen to look for those predicted off-targets, and if they're not there, that could potentially make it safer. You're never going to get a guarantee with anything, but there are ways to try and minimize it. And with recombinases, which are coming through now, which don't make any double-stranded breaks, potentially you could get away from that almost altogether.
Okay, thank you for that. So it's time now to go to our flash poster session. So thank you very much to our panel. So it's our final early career researcher flash poster session, and we were having some technical difficulties connecting with our first speaker. So Belinda, is Irina Savoya-Glazova now connected? Yes, I'm getting a thumbs up. Brilliant. So first presenter is Irina. She's online, and then we'll go straight to Maori after that. So Irina, hopefully you can hear me, and I'm handing over to you for your 3 minutes now.
Yeah, thank you so much for the presentation, Katerina, and hello everyone. So all we know that the main reason of cystic fibrosis development is mutations which occur in the gene coding for cystic fibrosis transmembrane conductance regulator. The CFTR channel is required for chloride transport and fluid balance. Just to remind you that the CFTR channel consists of 2 transmembrane domains and 2 intracellular nucleotide binding domains. And in our research, we are more concentrated on NBD1 region, since it contains F-avoid residue, and whose deletion is the most common mutation in cystic fibrosis. Next slide, please. Some time ago in our lab, we developed the library of nanobodies against human NBD1, and among them we found one which destabilizes the domain. This is T1A nanobody, and on the left side of the slide you see the graphic of thermal shift assay, and with the red arrow the destabilization of NBD1 upon binding of T1A is shown. By flow cytometry experiments, it has been shown that the cell surface expression of wild-type CFTR was decreased upon binding of T1A nanobody in contrast to the controls. This is the orange curve on the right side of the slide. And the analysis of crystal structure of the complex has revealed that the epitope of nanobody includes F-avoid residue, and all of these findings gave us an idea that T1A upon binding to NBD1 can lead to a similar destabilization effect as the mutation of FAVOI deletion. The next slide, please. So by means of hydrogen deuterium exchange experiments, we showed that some elements in NBD1 were affected upon binding of T1A to it in a similar way as ones which were affected by FAVOI deletion mutation. And probably these elements can be a part of the complicated allosteric mechanism which is needed for proper function of the domain. Also in our research, we use single molecule thread technique to study the dynamics of NBD1 landscape. And by means of this technique, we found that there is a special conformational equilibrium in NBD1 between so-called canonical and alternative states. So in our research, we hypothesized that T1A nanobody can mimic the destabilization effect of FAVOI deletion mutation, and it can be used as a good imitation to study the molecular basis of this mutation. Thank you very much for your attention.
Thank you. Thank you.
Yeah.
Hi everyone, I'm Mayuri Ratrat from University of Bristol. The triple combination therapy, elexacaftor, tezacaftor, ivacaftor, or ECI, has transformed the treatment of cystic fibrosis. Although the binding sites of ETI have been identified, its effects on the single-channel activity of F5-del CFTR have not been reported. Therefore, in this work, we aim to investigate how the ETI rescues F5-del CFTR channel activity. To do so, we treated F5-del CFTR expressing cells with ETI for 24 hours at 37 degrees C and performed single-channel patch clamp experiment using excised inside-out membrane patches. As a control, we studied F5-del CFTR rescued by low-temperature incubation. Here's the data. The recordings were made at 37 degrees C using excised inside-out membrane patches. Y-axis is current amplitude, and x-axis is time. And when studied at 37 degrees C, F5-del CFTR rescued by low-temperature incubation rapidly loses channel activity within 20 minutes. In contrast, ETI rescued F5-del CFTR demonstrates prolonged channel activity at 37 degrees C in excised inside-out membrane patches. These recordings lasted for 60 minutes, but others lasted even longer than 2 to 3 hours. Also, the ETI rescued F5-del CFTR can rescue more CFTR channels, as for example, in this recording, there are 8 active channels, whereas only 5 active channels are observed in low-temperature incubation. So, based on these data, we interpret these data to summarize, to suggest that the ETI rescued F5-del CFTR are stabilized at the apical membrane where they transport chloride for much longer. If you like to see the effects of ETI on how it rescues F5-del CFTR on individual F5-del CFTR channel, so please come to my poster number 4B. Thank you very much for your attention.
Hello, my name is Anna Veselu. I'm from the University of Bristol, where I'm using computer simulations to investigate ivacaftor-induced conformational changes in CFTR. Ivacaftor is licensed by the FDA to treat 97 cystic fibrosis causing mutations in CFTR. Jujen and colleagues identified the binding site for ivacaftor at the protein-lipid interface in a cleft formed by transmembrane segments 4, 5, and 8. However, the networks that link the ivacaftor binding site to functionally important regions in CFTR remain unknown. Thus, our aim is to understand how the ivacaftor binding site is allosterically coupled to functionally important regions in CFTR using computational methods. Our approach relies on molecular dynamics, or MD simulations. MD simulations allow us to predict how biological molecules move and interact over time. CFTR is a highly dynamic protein. Cycles of ATP binding and hydrolysis at the nucleotide binding domains gate the channel spore. We cannot understand CFTR's behavior solely through cryo-EM structures because they are snapshots in time. However, we can use the cryo-EM data to create MD simulations that then restore the dynamics of the protein. This allows us to learn how the CFTR protein reacts to drug binding. Using MD simulations of the phosphorylated ATP-bound human CFTR complex with ivacaftor, shown here on the left side, I generate a group of starting conformations for my second computational tool called Dynamical Non-Equilibrium MD Simulations, or DNEMD simulations for short. DNEMD simulations is a powerful technique that allows us to study how structural changes propagate through proteins. Specifically, shown here in the middle, I remove ivacaftor from its binding site to observe these changes. Then, I map the signal propagation from the ivacaftor binding site to identify the pathways connecting this site to functionally important regions in CFTR, such as the ATP binding sites. By mapping disease-causing mutations to these allosteric pathways, we can gain a better understanding of how ivacaftor restores CFTR function. If you want to hear more about how I ran these simulations and what they show about the signal transduction, please come find me at poster 4C. Thank you very much for your attention.
Hi, I'm Lucia from Paddy Harrison's lab, Cork. And today I'm going to present our strategy to correct G542X by gene editing. I hope you can see it's not too blurred. Through a collaboration with another group, actually from Italy, we have access to rectal biopsies from CF patients. And from these samples, you can isolate intestinal crypts that are commonly used to develop intestinal organoids, the same that Patrick showed earlier. And just to give you a little bit of context, those have been used to validate modulators, so they are considered as a reliable model. There are many different cell types within an intestinal crypt, but we're particularly interested into the stem cells, here in the middle of my slide. And our approach consists in transducing these stem cells with that thing in the middle that looks like a virus, but is not a virus. It's a virus-like particle that has been developed by another group and that we use as a delivery vector for our editing machinery. That is the one in blue in the middle. So they can bind the target cell through the glycoprotein and deliver this editing machinery into the cell so that it can get to the nucleus where the DNA can be edited. So if we zoom in into the nucleus, this is what we see. So we have our mutation G542X, and I won't get into the details, but what we use is called adenine-based editing. So we can edit adenines. And in this case, we are able, through the system, to get rid of the stop codon and go from G542X to G542R, which is another variant, basically, but it has been proven by other groups, such as David Bedwell's group, to be able to restore CFTR synthesis and function. So once we do the editing, the stem cells can develop into organoids. And how can we check if the editing worked? the organoids are like spheres, they are closed. If the editing did work, then chloride can go out, water can go in, so they swell. So in my poster, you can find QR codes that you can scan with your phone to watch the organoids as well. And if you have any questions, I'll be happy to answer my poster 4D. Thanks.
Hello, my name is Izzy. I am at poster 4E. So I'm also working with adenine base editing, and I'm actually using the base editor that Lucia has optimized to target G542X and convert it to G542R. Along with that, I'm also using some lovely lipid-based nanoparticles that are being developed at a group at UCL that Miriam's going to talk about in just a second. So combining these two, the vector and our adenine base editor, I'm then using ETI to help the CFTR, G542R, reach the membrane, and from there I'm doing electrophysiology, protein assays, mRNA, and also some wound healing scratch assays. My key finding that I just want to highlight is that with 17% editing efficiency, or 17% of all alleles edited, in addition to ETI treatment, we can restore CFTR function up to 60% of wild type when looking at Ussing data. And please do come and see my poster. Thank you.
Hi everyone, my name is Miriam Greenwood. And I'm a PhD student at University College London. My poster is entitled Nucleic Acid Delivery Using Nanoparticle Formulations to Treat Cystic Fibrosis. So one of the major obstacles to developing a gene therapy for cystic fibrosis is the delivery into the airways. So you have that thick, sticky mucus, which is very difficult to cross. And then once you've crossed that, the gene editing or nucleic acid therapy needs to cross through the tight junctions in the pseudostratified epithelium if they're to cross down to reach the basal cells, which lie at the bottom, which are ideal therapeutic targets because they're able to differentiate into all cell types of the lung epithelium. So many different vectors have been explored, many viral vectors, but they often trigger a strong immune response, and people develop antibodies to viruses such as AAV. So there's been a shift towards non-viral vectors, such as nanoparticles, which we've been developing to deliver the nucleic acid or CRISPR to rescue CFTR function. So I've been working with nanoparticles, and this is what Izzy used to transport in her CRISPR. So it consists of 3 different components. We have the liposome, which has 2 different lipids, and then a peptide. So the liposome is positively charged, and the peptide has a lysine tail, also positively charged, which have electrostatic interactions with the cargo to package that inside the nanoparticle. And then the peptide has a targeting sequence, you can see in green, which binds specific receptors on the cell surface of the lung cells for receptor-mediated endocytosis. And that's how the gene therapy enters the cell. So the aims and methods of my study were to explore uptake, GFP expression, and the inflammatory response to nucleic acid delivery of CFPE cells carrying the F508 deletion. So they're primary cells that have been semi-immortalized using an anti-senescent gene using lentivirus. And I looked at GFP plasmid DNA and GFP mRNA, and those were tagged with Cy5. So I could see by Cy5 by flow cytometry to look at the uptake, and then to look at GFP expression as a measure of transfection efficiency. And then I wanted to see if this whole transfection itself is causing inflammation, which isn't ideal in an already inflamed lung in CF. And I did that by measuring IL-6 and IL-8, two pro-inflammatory markers, within the supernatant using an ELISA, and their response to dexamethasone, which is an anti-inflammatory drug available in the clinic. For more information, please come to my poster 4F. Thank you.
Hello, so my name is Brogan, and I am working on developing biofilm models that are polymicrobial that more closely resemble that of the CF lung and how this can be used in therapeutic intervention. So, background, aim, and the model. So there's quite a few problems with models generally. There's no sort of general model that everyone would use. Everyone uses a model specific to what they want their outputs to be. So current models also only consider the monospecies, and they don't really consider how the CF environment can actually affect the therapeutic outputs. And also, we need a standardized model. So our aim was to develop a tri-species polymicrobial model that more closely resembled that of the CF lung that could be used for open access in academia and industry in the drug discovery pipeline. So this is the model. We do a modified colony biofilm model with tri-species, and it maintains high versatility, increased handleability, and increases the CF relevance. We grow the biofilm on a polycarbonate disc on ASM, or SCFM2, which are sputum mimics, and we can change the composition of this as we want, and it also allows us to vary our outputs. So we're able to do imaging, we're able to disrupt the biofilm, do colony forming units, metabolic assays, and also small molecule assays as well. So using my model, I've kind of focused on 4 main outputs. So I've benchmarked existing antibiotics, including first and last line meropenem and colistin. I've looked at effects of the CF environment. So we've done a range of this. We've looked at the effects of oxygen gradients, glucose in CF diabetes, and also how biofilm rheology can actually affect the antibiotic tolerance as well. Some of the really interesting data as well is looking at novel therapeutics. So really interestingly, we've actually got a paper in review at the moment looking at a ciprofloxacin conjugate, which actually shows really good activity against the whole biofilm, all 3 species. And we've also looking at a fungostatic against Aspergillus fumigatus. And finally, because everyone loves pretty pictures, I do a lot of structural and imaging analysis where I've been able to show some really nice images about how they interact and how they associate with each other, or how they move depending on the conditions and the treatments. Some of the really nice conclusions using this model that I've been able to develop is I've been able to develop a highly amenable polymicrobial model, which is suitable for increasing complexity. So I've been able to add Burkholderia, and I've also been able to add Aspergillus. And I've been able to show that these polymicrobial biofilms actually do increase antibiotic tolerance, and that the lung environment of people with cystic fibrosis can actually affect antibiotic tolerance as well. And this is both antibiotic dependent and CF environment dependent. And finally, I would just like to thank Miguel Camara and everyone that has to put up with my incessant moaning and talking, because I talk forever. And so if you'd like to know any more, because I will talk for hours about this and I have got plenty of pretty pictures to show you, please come and find me after or at my poster. Thank you.
Thank you very much. So we won't be having a presentation for poster 4H. So if you get an opportunity to go and have a look at Holly's poster, it is out there, and I know she'd really love you to see it. So we're going to start to wrap up, but I want to say thank you so much to all of the early career researchers. It's a really big deal getting up here and presenting in front of all of these people. I know for many of you it was the first time, and honestly, you wouldn't have known that. It was terrific. So thank you. Extra round of applause for them. And moving seamlessly on, there are obviously prizes to now present. So I'm going to ask Lucy, and I think, David, you're coming up as well, to announce the winners of the poster judging and to present some certificates.
Hiya, just to echo Catherine's comments, thank you so much to the early career researchers for being part of the day. It's been absolutely fabulous. I've loved these flash poster sessions. We've had some really, really positive feedback as well from people about having the posters there, being able to talk to you all. So thank you very much for joining in and taking part and putting up with the pressure of having to come and talk up here. So I've got some prizes or certificates, I'll downgrade that, certificates to give out for the posters. So we've got Best Summer Student Certificate and all of the summer students will also get certificates, so please do wait around at the end of the session, and I think we have some tote bags as well that Belinda will be handing out, so make sure you find Belinda at the end of the session. And then I've got first, second and third poster prizes for the early career researchers, and David's going to shake your hand. Okay, so we will start with the Best Summer Studentship Poster Prize, and this goes to Jasmine Johnson. Okay, so in reverse order for the Early Career Researcher Poster Prize, we have in third place, Mayuri Rodrat. Well done. In second place, we have Isabel Rose. Well done, Isabel. And in first place, we have Ellie Slater. And so I'd also just want to take this opportunity to thank a few people before I hand over to one of our trustees to do the closing remarks. So in terms of thank yous, obviously thank you very much to our sponsors. So we have Vertex, LifeArc, Nordic Pharma and Optics 11. Thank you so much for your sponsorship. Without that, we wouldn't have been able to put this event on. I also want to say a huge thank you to the Outlook team who sat at the back and around the room and have helped put on this event. They've been fabulous throughout. They've done a great job. Again, thank you very much for all of your support. We really do appreciate it. In terms of the Cystic Fibrosis Trust team, obviously thank you to the whole working group. Specifically, I'd like to thank Natalie, Nicola, Belinda and Paula. You've done an awesome job herding lots of cats, and we really do appreciate it. Thank you very much. I just want to say thank you to all the staff here that have volunteered from the Trust to come and help today, all the venue staff. The venue have been fantastic with us, actually, because we had to make a few last-minute changes to the event that was supposed to happen tomorrow due to various train strikes. So they've been really accommodating. So thank you very much for that. And I think just thank you to all of you for joining in and participating, those of you in the room, those of you online. From my perspective, it's been a fabulous event. There's been so much chatting and conversation happening outside the room, which is absolutely fabulous to see. I hope you've all benefited from that and come away with some new research ideas or new partnership ideas. And I'm now going to hand over to Michelle Shaw, who's one of our trustees, who's going to provide a few closing remarks.
Thanks, Lucy. Oh, what a lovely time to join. Well done, everybody. So I wanted to say good evening, everyone. I'm joining you remotely from South Yorkshire. I'm hoping that the technology will hold up. I'm sure Lucy will let me know if it's not. I'm Michelle Shaw. I've been a trustee of the CF Trust for 4 years now, and I have joined the conference every year as a delegate online. What struck me today is that there's been a really warm collaborative feel to the day, and I hope you have felt that in the room because it has come across online. I think it's partly the emphasis of the James Lind research priorities, partly because of the multiple funding partnerships, the CF centres working together, the collaborative approach there, and the level of involvement of the people with CF in decisions about research. I think all of that has come together to create a really clear, community-guided feel for the day. Lorna used the word teamwork, which I thought was perfect phrase. And this is really good in the current environment because I think at the moment, almost everybody's experience of CF is unique to them. Everybody's experiencing it slightly differently. As a trustee, you're very mindful of the hard work, the challenge and the dedication that has gone into fundraising. And you feel an intense sense of duty to ensure that those funds are used wisely and in accordance with the CF community's wishes. And the James Lind Alliance Research Priorities Refresh has provided a really good tool for the trustees in doing that. As well as being a trustee, I'm also a person with CF. And this year I've been involved in some of the research engagement activities that Lorna was discussing earlier. And I absolutely agree that this is a really important step forward. It allows people with CF the opportunity to feel empowered and to give them a positive feel about the future. I'm in that very small minority who can't benefit from the modulators. So for me, the involvement felt like I was given a voice. I also think it's important for those strategic partners who are hearing our input because they get a real sense of what it's like to have CF and what the experience is being involved in clinical trials. And I don't think they would have otherwise had that. There has clearly been a lot of exciting progress since the last CF Trust conference, and that has really come across today. And I think this event is really good at sharing that. I particularly enjoyed the question and answer sessions. It's really hard to describe the feeling that you get when you listen to very experienced, dedicated, accomplished professionals talk about things that really do affect you on a daily basis. And they discuss it with enthusiasm, with an aim to manage or conquer those problems. Also, it was great to see the poster presentations. I think they did so well, fitting in so much information in 3 minutes. I really am impressed. Jane made an observation earlier this morning about trying to get to clinicians and researchers at the earliest possible stage in their career. And that made me smile for 2 reasons. First of all, it's so true. As people with CF get older, so do their doctors. I constantly find myself saying to myself, be nice to junior doctors, be nice to junior doctors. I'll be there in the future. But also, it reminded me of my daughter. My daughter's 9, so I don't think it's exactly what Jane had in mind when she made that comment. But recently we've been talking about medical research, and it started because she heard the phrase, medicine's discovery catapult. And to a 9-year-old, I'm not sure what she had in her head, but her response was, wow, what's that? So we had a conversation, and then that moved on to gene therapy because they're learning on a very basic level about DNA. Yesterday, after having a couple of conversations, she told me that she had put herself forward for the role of Minister of Science at the Pupils Parliament, which is a local scheme, and she'd been shortlisted. And one of the reasons that she'd given for applying when she was interviewed was, science is amazing and school children should know how important it is in real life. Oh. So my daughter aside, I know Lucy and her team have put a lot of thought into attracting early career researchers. So I'm really hopeful that we'll see the rewards of that in future CF conferences and in future years. And I'm really glad that there was such a good turnout today. So to conclude, I'd like to thank everybody who's been involved in the conference today and everybody who's joined online. And to the researchers and the clinicians, please keep going. It's really appreciated. We hope that you've been motivated as well today, and we really do hope we see you next year.
So thank you very much to Michelle. It's now down to me to formally close the event. Just speaking personally, this is the third time I've been involved in this conference, and it's always such a pleasure to facilitate. I really enjoy the experience of being amongst your community, and you know, what stands out to me is the determination and passion of every single person in this room. It's always impressive, it's always inspiring, and I was really struck by the way in which that determination and passion is embodied and is continuing with the 23 early career researchers and the 5 summer students who we met today. So, you know, I guess similar to what Michelle has said, I hope that you're feeling, particularly those of you living with CF, I hope you're feeling a sense of optimism about that. In a moment, I'm going to ask the poster prize winners to come up for another photo, and also the summer students, if you could come up to the front as well. I'm going to ask everyone else in the room to go back out to the foyer for the drinks reception and to continue to view the posters. But for now, we're going to end UK CFC 2023 by saying thank you so much, everyone, and hopefully see you next year.